I am very glad to have your attention on this, and today we are having to discuss the G6PD deficiency and glutathione association. So what relationship exists in them and what significance do they have in hemolytic anemia? To begin with this discussion, I would start to say that these particular steps we are coming to discuss occurs within red cell, and the reason is that all other cells that do have membrane-bound nucleus and mitochondrial matrix undergo the respiration, which is the aerobic process of energy production, ATP production. But red cells do not have matrix like mitochondria, and they do not have nucleus. Therefore, even if they go through the glycolytic process and they produce pyruvate, it cannot take the pyruvate derivative into Krebs cycle or the tricarboxylic pathway, which means it cannot produce energy in that pathway. Now, the commonest process by which red cells produce ATP is the prominent Embden-Meyerhoff's pathway. The 90% of ATP production in red cells is through this Embden-Meyerhoff pathway. There is one side process which is just 10% of it, and it is named as the Hezos monophosphate shunt or the pentose phosphate pathway. Now, it is directly associated with hemolytic anemia, and the interesting aspect is we already know from this first discussion, I just want to bring it up here, that in the first stage of glycolysis, glucose is converted into glucose 6-phosphate, which is G6P, by an enzyme called hexokinase with the use of ATP. And of course, hexokinase is one of the enzymes that we say it is ubiquitous. It is present in all cells that do produce ATP or undergo glycolysis. So red cells do have the hexokinase, which is able to facilitate the conversion of glucose to glucose 6-phosphate. The rest of the steps were supposed to undergo an Embden-Meyerhoff pathway. However, the only process by which red cells are able to produce the NADPH, which is the nicotinamide adenine dinucleotide phosphate reduced, is through a glucose 6-phosphate dehydrogenase conversion of glucose 6-phosphate into ribulose 5-phosphate. An interesting step here. Just keep this and we will go directly into how relevant is producing this NADPH by this slight 10% chance has an effect on matured red cells. I will take you back onto this. This is the first one I'll take you onto. We discussed that this is the reduced form of these coenzymes, and this is the oxidized form. And again, I mentioned that the reduced form are in a full state. It means they are occupied, which they can't receive, but they can only donate electrons and release protons. Whereas the oxidized states are in the state of hunger, where they can receive electrons and protons. Lovely. Now, the next thing I will take you back on is when I was explaining about how the lipoic acid is the active site disulfide bond here on lipoic acid makes it able to receive the hydroxyethyl TPP complex. Now, I said one, the disulfide bond is the oxidized state of lipoic acid, which means it is in a state where it can receive this complex. 2, I said when the lipoic acid interacts with this hydroxyethyl TPP complex, it's the electron transfer between it causes it to lose one of these sulfur bonds, and therefore it becomes an SH instead of SS, and that SH status is what we call the sulfhydryl bond. The sulfhydryl bond means that it is not in its full state of oxidation, or it is not in its full oxidized state. It is partially oxidized, and in that state it can partially activate, or it can partially interact with other motifs or other compounds, but not its original compound here. Great. And I said whilst it interacts with the acetyl group, it then undergoes another electron transport, which then loses the second sulfide sulfur on here, making it now a dihydrogen, meaning it has no sulfur on active site. Lovely. So we can then rush straight forward to discuss about glutathione. We are on this page now. I will begin with the explanation from these four corners, because you already know what a reducing agent and an oxidizing agent is. So putting much of your focus on here. I have explained that this is disulfide bond. This is partial sulfur and hydrogen bond, which is the sulfhydra. And this is the dihydrogen bond, meaning all the active site has been replaced. Now, this is glutathione peroxidase. An oxidase enzyme, or a peroxidase, makes an oxygen active, or it enhances the reactivity of oxygen. It helps to incorporate oxygen into a compound or a reaction. 2. It facilitates the transfer of electrons or protons. 3. It is highly responsible for detoxification within a biological environment, or wherever it is able to do that. Now, glutathione. GSH is glutathione sulfhydra. It means that this glutathione has an H at the end, which means it is in a state of reduced. And lastly, it means that it is in a state that it can donate electrons. Now, that state is the SH state of glutathione. We called glutathione sulfhydra. On the other hand, this is GSSG. This is a glutathione that has got a disulfide bond. And already I've explained previously that when you have a disulfide bond, it means that that particular compound, that particular subgroup, that particular coenzyme is in a state where it can receive, because that is the active subunit of glutathione, which is the disulfide bond. So at this stage, it can receive, it can bind to, it can accept, but it cannot donate any protons or electrons. So that is the GSSG that we have here. Now, when this GSH reacts with hydrogen peroxide, superoxide, hydrozonium, nitric oxide species, like a reactive oxygen species, it loses the hydrogen, and therefore we have GS. So for instance, if GSH and this is GSH, when both of them undergo a reaction with reactive oxygen species and they lose their H, we have this H off, another H off, and we are having left with GSSG. So understand that in this state they are unable to donate protons because they have lost the H and H. That's why I put these two together, so that you will know why we get the GSSG. Is when the GSH, the reduced state, react with reactive oxygen species and loses this proton, which is the H plus, it is left with a GS, and then another GS react with the GS and forms GSSG, meaning they are in a state that they can't donate proton, and at this stage they can only receive, and we call it oxidized. This is very meaningful and you should know. When you look under glutathione peroxidase, this is a peroxidase, like I told you, it transfers hydrogen into a reaction to allow detoxification. Now, glutathione peroxidase will catalyze the transfer of an H from this reduced glutathione to a hydrogen peroxide, which is a potential oxidant, which can cause oxidative stress to red cells, to convert it into water, 2 molecules of water, or sometimes into water and oxygen, whatever it converts it to, so that it changes its state of potential oxidant or as an oxidative stress to a non-toxic or a non-harmful product. Now that you know what glutathione does, and secondly, you know how glucose phosphate helps us to obtain an NADPH. The process here is This is the only process by which red cells obtain NADPH, which is the reduced form, through the glucosase phosphate dehydrogenase conversion of glucosase phosphate into ribulose 5-phosphate. So in between these two, there is a release of the reduced nicotinamide adenine dinucleotide phosphate from glucosase phosphate dehydrogenase. This is an enzyme. Now fast forward, I've explained to you that NADPH is one of the most stable coenzyme responsible for electron balance, and I have explained to you that this is the oxidized state, a state where it doesn't have protons or H+, and it is a state that it can receive. The glutathione reductase is a reducing agent. It is going to reduce the glutathione oxidized into a reduced agent by transferring a proton from NADPH to one of the GS. I have told you this GSSG is because it is a GSH that has lost the H and another GSH that has lost the H, so they come together to form GSSG. So when one of the H here is transferred to this GS, it makes it a GSH, which is the reduced form of glutathione. So even as it transfers the H from the NADPH to the GS, making it GSH, the remaining is the NADP without the H. So in this state, the glutathione reductase has been able to catalyze the transfer of an H, a proton here, to the glutathione oxidized, and making it reduced here for the next cycle of detoxification by glutathione peroxidase. Now our interest comes down here. Now you know how all this process works. Where are the sources of reactive oxygen species? These include superoxides, hydrogen peroxides, nitric oxide species, hydrozonium ions, hydroxyl radicals. Some sources include the red cell oxyhemoglobin autooxidation. This is what I mean. The red cells are hem proteins. They contain iron 2+, and they transport oxygen. In their transport process, sometimes they get overwhelmed and they are auto-oxidized, meaning at the moment when you are oxidized, you are already 2+. When you are oxidized, you gain more pluses, which means you lose more electrons. And at that stage, it becomes an Fe3+, and whatever red cell, matured red cell that contains this F3+ is a methemoglobin, which cannot transport oxygen, or which has difficulties with release of oxygen. And in this stage, if a methemoglobin reacts with a superoxide, there is a dismutation that forms hydrogen peroxide. So this peroxide is actually a superoxide, and the dismutation creates the hydrogen peroxide on here. Sources of reactive oxygen species include tobacco. Let me do this correction. Yeah, so I've changed it to superoxide. That was actually a typo error. So that's tobacco smoke, radiation, and some external pollutions can also create reactive oxygen species. So when we talk about G6PD deficiency, the end result is that if there is no glucose 6-phosphate dehydrogenase, so if this is not, it means that 1, this NADPH is not produced. This is one of the strongest electron balance in the biological system to receive electron transfer protons and all that. It means that this will not be produced. Good. If this is not produced, glutathione reductase is not going to get any hydrogen source to transfer to the oxidized glutathione, which means that the reduced glutathione will not be formed. And it goes on to say that this H+ will not be transferred on by the glutathione peroxidase. So the glutathione peroxidase will be there, but it cannot obtain this H to transfer to hydrogen peroxide, which means that there will continue to be oxidative stress. This one is not cancelled, it is pronounced. High level of oxidative stress, radicals being produced, putting stress on your red cells and causing them to hemolyze. What are the clinical appearance? In that state, the patient will continue to hemolyze the red cells, and they will run anemic, being very weak. And the G6PD deficiency is tested in pregnant women for early detection of possible neonatal jaundice because of the hemolytic anemia, to estimate a pregnant woman's chances of suffering preeclampsia. And of course, it is X-linked, the gene that causes G6PD deficiency. So G6PD, that enzyme, is produced from a gene on chromosome X. So if it is on chromosome X, it means that females have XX chromosome. If one of the X is suffering, the other X is working. But if the man has chromosome X mutation or any defect that causes malfunctioning or malproduction of the G6PD enzyme, man is doomed because he has only one X chromosome. Anyway, that's life. And the last thing I'll say about dose pen is that it is tested in pregnant women. It also gives them an idea what kind of drugs that they are supposed to give to the pregnant woman or whoever is being tested, because some drugs causes oxidative stress or causes stress on your blood cells or production of some reactive oxygen species. Some drugs predisposes you to haemolytic episodes. So just to have, for instance, sulfur containing drugs have some very significant effect on that. Fast forward our last slide and we go. I can't conclude a discussion on G6PD without introducing you to something very interesting. So if somebody has glucose phosphate dehydrogenase deficiency, the possibility of them having their red cells looking this way is very high. But you can see them on your blood film, and you can skip them when you don't know them, or you can misdiagnose or miswrite some stuff when you don't know what it is. So the difference of these cells is this is hemighost cells, which means the cell looks ghosted. All these cells are red cells that have got some of them being normal and others having defects. If you look at these red cells, they look quite normal. We can't talk about their size because there is no lymphocyte nucleus to compare here. But if you look at this cell, you can see that all the other sides have stained. They are hemoglobinized except a portion of it. So there is a defect hemoglobin within this red cell because of the oxidative stress, and it has denatured the hemoglobin in this portion. So in this case, when that red cells undergoes a staining, the denatured portion will not be stained and they will look white. It doesn't mean there is a hole or something. It just means that that portion is not stained, but that is how it will appear under your microscope. But the difference here is that it has an outer cell membrane outline that you can see. That's why we call it a ghost. It looks there and not there. That's why we call it a ghost. And it is these hemighost cells, if it is in the peripheral blood and it's going through these micro paths within the blood vessels, different vascular layers, there's that possibility of it being hemolyzed when it comes in contact with vascular surfaces or any very compressed areas within the vascular endothelium. It can just hemolyze. That's when we get the blister cells on there. And looking on here, let's see, we have bite cells or dermatocytes. So these bite cells are as a result of Hen's bodies. So I will explain these Hen's bodies first. Hen's bodies are denatured hemoglobins that have affected the cellular membrane. So this denatured hemoglobin extends to the surface of the red blood cells and form a purple shaped spot which is insoluble, and you can see a small spot on the surface, and then if you look up here you can see this purple so it is actually on the surface of the red cell, and those are what we call Hen's bodies. And Hen's bodies are denatured hemoglobin, not ions. They are denatured hemoglobins. And when the macrophages within your spleen finds them, they see them as an abnormal thing on a normal cell, and therefore it bites it off. And when the macrophages in the spleen bite it off, it looks like this, and we call them bite cells. So when you see bite cells, you see Hen's bodies and you see hemighost cells within the peripheral film, start looking for relevant approaches to help your patient and know the what these things mean, the possibility of hemolysis in there, check the HP, check patient history, and you know what to do to help your patient. It is a very lovely time having you on here, and series of these kind of educational videos will be coming up. If you love this, how meaningful it is for you, you can subscribe to this channel, you can like it and share, and you follow me on YouTube, Facebook, and sometimes I go and play on TikTok. Thank you very much. Have a very nice day. Goodbye.